Hi, Karin, and welcome. A few years ago, there was a lot of talk about cytokines in Denmark and the relation to fibromyalgia, and maybe it could be a diagnostic tool. And then I looked into it a bit, and I can see that you've done some work on proteins in blood, more general, not just cytokines, but in general. And I would like to discuss with you what are actually proteomics. We'll get into that. And why is this interesting to look at when we talk about fibromyalgia? The reason why we are able to do this is because we, the Fibromyalgia Association in Denmark, we got a grant from Helsefonden to do interviews and talk about research to a audience that doesn't have a research background. So thank you so much for being here, and maybe you can introduce yourself.
Sure. Thank you so much for the invitation, Leah. It's always fun when someone reads your thesis, even though it's getting 4 years old by now, but it's always fun when you get emails that someone has read it. So my name is Karin Wåhlén. I just want to start to say that I'm not a clinician. I'm just a simple medical biologist that have a background in biomedicine. And at the moment, I work as a lecturer in experimental pain and rehabilitation medicine at the Department of Health, Medicine and Caring Sciences here at Linköping University, and I belong to the Painomics Research Group. And my research journey kind of started 10 years ago when I did my master's thesis, where I worked with proteomics, but in another field than long-term pain. And after my master's thesis, it opened up an opportunity to join the Painomics Research Group as a research assistant. So I took that one. And by then, I didn't know anything about pain, but definitely not anything about long-term pain or the diagnosis of fibromyalgia. So I've really learned a lot the past 10 years. And what was striking when I started was that when it comes to biomarker research or proteomics in general, that very little is known about the underlying cause of fibromyalgia. And nobody had really used this kind of methods that we are experts in Painomics Research Group. So it was super interesting to start to work within this field.
Maybe you can explain just what are proteomics actually, and what are proteins in your blood? I mean,
sure, I can try at least.
Yeah.
Yeah. Okay. Let's start with proteins. You know, proteins are like essential molecules for living organisms. Proteins can help to build and repair tissues. It can control chemical reactions, send signals between different cells in our body, and so much more. And a lot of our proteins are circulating in our blood. And our body consists of different types of cells and tissues, like skin, muscle, the brain, immune cells. And all these tissues and cells has their own sets of proteins that helps out with different type of functions. Proteins go from very, very small molecules like cytokines, and cytokines are more involved in immune cells that for example, when you have inflammation, your immune cells express different kind of cytokines to recruit more cells to take care of an inflammation or a damage, for example. And the terminology proteomics is just like the study of the complete set of proteins in a cell, organism, tissue, or biological fluid at a specific time point. And I tried to find a good layman term how to explain proteomics, and I found one that you could, let's say like your body is an orchestra and proteins are the musicians, and each protein, which is the musician, has a specific role to play in this orchestra, and together all these musicians create some kind of symphony of life, how the body functions. Proteomics is the study of all these proteins at once, understanding how they work together, how they can change in different conditions, and what happens when things go wrong. And if someone is is healthy, then they all play in harmony, for example. But if someone is sick, maybe these musicians or proteins are out of tune or are missing. And that's what we're trying to discover. And by studying like all the musicians or proteins in a present sample in a biological fluid, for example, then we can figure out how these conditions are changed during different disease settings, for example. And that's why it's so important when you study this kind of cohorts that you also have a reference group like a control group, so you know that you have a healthy reference protein to compare with. And then in 2016, I got the opportunity to engage in a PhD or in my PhD studies. And the aim of my studies was like to investigate how the molecular protein pattern in blood differed between patients that had the diagnosed chronic widespread pain or the diagnosed fibromyalgia, and then compare it to healthy controls. And as you know, we also have a lot of like clinical characteristics on our patients cohort. Where they have filed a lot of questionnaires where they report their pain intensity, how sensitive they are to pressure threshold, for example, or like psychological stress. They report anxiety, depression, and fatigue and stuff like that. So we wanted to also investigate if these clinical characteristics in our pain cohorts had a correlation or a connection to the discovered protein pattern in blood. What we saw in my 4 studies combined, if I should try to summarize it, is that we can see differences in the expressed proteins in blood in our fibromyalgia patients compared to healthy controls when we just look exploratory. And the proteins that we saw that were changed were related to different kind of processes in the body, such as immune system, inflammation, and metabolic processes, very large general processes. What our data also suggests is that we have specific protein profiles that are related or correlated to specific clinical characteristics, such as pain intensity. You know, when they rate their pain from 0 to 10, and 10 is the worst imaginable pain, we can see that if you had a higher pain intensity, you had a specific protein profile related to specific processes. So that was also the case for other clinical characteristics, such as anxiety and depression, that is very common in long-term pain cohorts. We could also see that exercise is good for reducing common pain symptoms, and it of course improves your muscle strength. So exercise is really good for you, and also to some extent resistant exercise could affect the protein content after 3 months, but that's just as an overview. So this exploratory studies that we do when it comes to proteomics are pure basic science. I mean, we're trying to figure out disease mechanism in long-term pain, as fibromyalgia is, because we don't know how it works in the body yet. So that's kind of the overall aim why we use proteomics as a method in fibromyalgia. And I mean, this pattern that we and other researchers has identified gave us more a hint on what kind of hypothesis we need to go further with and explore specific protein pathways, for example.
Based on your findings from your studies, is there specific types of proteins that are interesting when we talk about fibromyalgia?
You want to know if we have found any biomarkers or?
Yeah.
You know, I just, before we go into like specific question about that, I just think it's important to talk a little bit about the method, because we have targeted analysis. Let's say you are a fisherman or fishing woman And you're sitting there with your rod, with your specific bait in this big lake, and the lake could be the blood, for example. And then if you're lucky, you're able to catch a fish with your bait, for example, a specific cytokine. And that one is like more a targeted analysis, then you know what you're looking for. In the untargeted analysis, the fishermen or fisherwomen catch out this big fishing net, and we catch everything that we can find in this big lake. And we analyze everything, all the proteins, different kinds of sizes and functions and stuff like that. And then we compare it. What do we see in fibromyalgia patients, and what do we see in healthy controls? And do we see any differences, for example? And that's more like the gold standard to do in proteomics, this untargeted analysis. And depending on which method you use, you of course catch different types of proteins. So what's important when it comes to proteomics is to combine different kind of fishing nets to be able to catch as many proteins as possible to get a comprehensive picture of the protein profile. The methods that we have used in proteomics, there we don't really look at cytokines because they're small. Then we look at larger proteins that are involved in other type of processes. But maybe we can talk about trends later, but it's very trendy to analyze cytokines at the moment and call it proteomics, but that's not really proteomics, because proteomics, it's more an untargeted approach where you don't know what you're looking for. The cytokines is known, then you know, okay, we're interested, for example, in IL-6 and inflammation, and then you go for a targeted analysis. But for proteomics, it's more untargeted analysis. You don't know what you're looking for, so you want to catch all the other type of proteins. But cytokines is, of course, important to look at in fibromyalgia. And then back to your question, if we have any specific protein types that we have found in our studies. Okay, so what we see in our proteomic studies of fibromyalgia and CVP is that we can see that proteins differ in expression. They go up or down in our patients group compared to our healthy controls. And that has other research have seen that too. And we don't look at a specific protein. We try to figure out all this protein that we see that we have catched with our fishing net in our big lake, which is the blood here. What kind of processes or networks are these proteins involved in? And
so it's more a pattern than a specific protein.
Yeah, because I mean, all proteins work in like harmony together, or disharmony, if you can say it like that. So we will never ever look at only single proteins, because we know that can't explain the whole biological pain mechanism behind long-term pain. It really doesn't work like that.
No.
So it's more about trying to find this, we call it protein disease signatures, like look at the pattern. Okay, we see that these protein profiles are involved in changes, for example, in the complement system that are involved in the immune system. Okay, we see differences, but we don't know what it does. So we need to go further to explore, like, okay, what these specific patterns that we see, what kind of function do they have? And that we don't know yet. So since it's just a exploratory study.
I assume it must be really complex, because a lot of things can change your pattern of proteins, right? What you eat, what you drink, if you exercise, where you're born, maybe?
Absolutely. There are a lot of factors that could change the protein expression. That's why we need to use standardized protocol when we do the collection of our biological samples, and when also we need to have the same type of methodology when we do the analysis. But because just preparing the sample could also affect the protein content. So that's why we follow strict protocols. But there is no consensus when it comes to how and when we should take the samples, or which method we should use, unfortunately. But the trends that our researchers are showing is that, okay, we see the same type of protein processes that are up or down regulated in fibromyalgia. So it at least tell us something or give us a hint on what type of like pathways we need to go further with. But what's also we need to remember that these are very, very small cohorts. So we can't say anything about that we have a biomarker or this is the truth, or we haven't figured out what's causing fibromyalgia when it comes to the molecular pattern yet. It just gives us a hint where we can go in the future.
So it's a step on the way to maybe figure out what's going on when you have fibromyalgia.
Absolutely. So, I mean, proteomics is more trying to understand like expression, function, and regulation of the entire proteome. And what is actually the entire proteome? Here it's limited to the type of method that we use or type of biological fluid, but we also need to combine results from different kind of body fluids, because blood is one thing, and then we have maybe saliva is important, then you have the cerebrospinal fluid, the fluid that is in the brain, and then you have the muscle tissue, because we know our pain patients has pain in their muscles, so maybe it's interesting to investigate that. So it's more about combining different kinds of all information that we get to get this comprehensive full picture of how it works.
What is the relation between proteomics and DNA? Does your proteomics depend on your DNA, or can it change?
So the DNA codes for our proteins, so it's definitely related in somehow. So it's of course important, like in the future, to also look at the genomics, which is the study of DNA, to see if the DNA changes, like mutations in DNA could affect protein expression, definitely.
Okay. My next question, you have answered it a little bit, but I'm going to ask it anyway. How far is proteomics from being a tool you can use to diagnose if someone has fibromyalgia?
You know, I wish we could say that we are already there, but in reality, proteomics for fibromyalgia is just in its infant. We have just started this field compared to other fields such as cancer or cardiovascular diseases. And I would love to say that we have a biomarker for fibromyalgia, but unfortunately we don't. And to be honest, I'm not sure that proteomics can be used it will never be used in the clinic, to be honest. I don't think so, but we can use proteomics as a tool to develop new tools in the future. For example, when we know how the underlying biological mechanisms work for long-term pain, then we maybe can develop new targets or look at more panels of proteins. But I think we're super far from that, and we don't have like a biomarker for fibromyalgia, unfortunately. But I mean, this research is super important. It's basic research and it's driving the field forward. So that's why we do it, trying to understand why.
Could you see proteomics to be a tool in understanding what are the things that everyone with fibromyalgia has in common and how do they differ?
I think proteomics is a valuable method in basic research to try to figure out what's going on in the body and trying to really understand why we get long-term pain. But I also think that proteomics can be used. You need to combine it with the genomic studies, of course, but also with all this available clinical characteristics, the symptoms. We still need to listen to our patients when it comes to their background stories and what type of symptoms they have, to be able to get the comprehensive pictures. And of course, maybe subgrouping patients in the long term, but I mean, we're quite far from that now. Yeah.
I wanted to ask a little bit about, you mentioned that you did some research into exercise and fibromyalgia.
We first analyzed the plasma or the blood proteome before start of intervention, and then 3 months after interventions. And then we compared, could we see any differences in any type of the proteins changing at baseline between fibromyalgia patients and healthy controls? And did this change after exercise. And what we did see, we saw to some extent that some proteins that were involved, if I remember correctly here now, in, for example, immunity processes got altered after exercise, but it wasn't to that extent that we can say that exercise reduced these proteins and it's good for reducing pain. We can't say anything about causality, for example. And once again, it's a super small sample size. What we saw was that, okay, exercise is good for our patient, those that are able to perform it, but maybe it's not suitable for everybody, because we didn't see that all proteins that were, for example, high in inflammation were super reduced. It was more a mix of protein patterns that we could see. But also what we saw in this in fibromyalgia group was that we had different subgroups of patients, those that had, for example, in the beginning, better muscle strength, for example, or lower muscle strength. And though if they then exercise for 3 months, then of course you can assume that those with lower muscle strength will build more muscles, or those that already have a good strength in the muscle were able to continue more throughout the study, and that could influence the differences in proteome. So predict is maybe a strong word, but I think that we can see patterns when we look at the molecular profiles. Prediction, maybe in the future, we can use proteomics to evaluate if an intervention works good or not. But we're definitely not there now.
I think lastly, I would like to talk a bit about the future and what you're doing now and would like to do, and where you see the research going. So do you see, or what are in your opinion, the most important trends in this research in fibromyalgia now and maybe in the next 5 or 10 years? Where do you hope that we're going?
Well, I mean, as we talked about, proteomics for fibromyalgia is in its infant. It's just in the beginning, starting phase, this discovery phase. What do we see compared to healthy controls, and can we identify subgroups, and can we relate it to clinical characteristics and stuff like that? What I see it's important, it's not to continue screening thousands of pain patients, because that only maybe confirm what we have seen further. What we do need to do now is that, okay, we have seen this disease pattern, if we can call it that, in fibromyalgia. We need to go, like, do back translational research, go to see, okay, go back to animal and cell studies to analyze, okay, these proteins or networks that we see, how does this work in animal models, to really figure out their function and expression. And when we know that, when we kind of have confirmed that it's true pain mechanism, for example, then we can go back to screening, and that maybe in 5 to 10 years, then we know that. I think that is really the future and the way we should go when it comes to proteomics and fibromyalgia.
This has been so interesting, and I've learned a lot. Thank you so much for doing this interview. And
Thank you so much for the invitation.
You're welcome. It's been a pleasure.